We were just sitting in our office the other day, and I just looked at it and, wow, I can't wait to catch grasshoppers this summer. We had a long, cold winter, rainy, or not that rainy winter, well, recently it has been. But they've done so much work collecting grasshoppers, analyzing the data, and I'm so happy that they get this opportunity to share the work that they've been doing. And so our first speaker today is going to be Trevor Holt. Trevor is a true jack of all trades. This guy, depending on what time of the year it is, he's either going to be making some kind of, what was it, elderberry syrup? He's making homemade chocolates. He's writing a novel. Whatever it is, Trevor is doing it. He's doing it well, and he's doing it in a way that other people aren't doing it. One of the most creative students I've ever interacted with. And so it is my pleasure to welcome up Trevor Holt. We're talking about grasshoppers today. Please join me in welcoming him.
Hi, thank you all for coming today. My name is Trevor Holt, and I'm here to talk to you about the grasshopper biodiversity of the Cascade-Siskiyou National Monument.
I can't see you.
Right here.
So,
Huh?
because here's the camera.
Yeah.
So you could go behind the desk too.
And first, I want to talk about what causes biodiversity decline. Now, there are two potential methods that can cause biodiversity to start to dwindle in an area. And one of those methods is climate change. And as you can see, this is a big old heat wave that we experienced in the West Coast in 2001 or 2021. And in this heat wave, a lot of organisms throughout the West Coast experienced these temperatures, and they had to adjust to the change. Some of these were ectotherms, like grasshoppers, and these grasshoppers started to proliferate exponentially, and we saw a lot of that here in Klamath. There's also human development that also impacts these ecosystems and causes them to causes us to encroach on them, and sometimes making these grasshoppers to move around us, move around what we are doing in the monument and across the country. So this is an article from the same year where grasshoppers in Klamath were having plague-like numbers, and that's likely due to the heat wave. These climate irregularities can cause misalignment with all species, especially species that are general specialists, and they require specific niches. The niches can be kind of hard to explain until you see it. So essentially, we need to do a lot of research to understand these grasshoppers so we can better monitor the environment. There are generalist species that can have a very hardy personality, and they can experience a wide range of temperatures, eat a wide range of food sources. And if these temperatures rise with ectotherms, being grasshoppers, they experience these temperatures and they take in the energy, the increased energy from the higher temperatures, and they will lead to lots of problems, and they can invade other places and cause declining in biodiversity. With human encroachment, we have cattle grazing across the grassy plains, which is where grasshoppers are most abundant, and the farming that happens in the Klamath can cause increased temperature or cause increased food source for certain grasshoppers that like what is being farmed. There's construction throughout private property throughout the monument, and if that private property isn't managed properly, it can be detrimental to many species throughout the monument. These power lines create grassy land highways for many grassland species to potentially move throughout the monument, move outside the monument, and come into the monument from foreign areas. This is right through Hobart Bluff. By monitoring grasshoppers, we can monitor the ecosystem. They are the most abundant grassland herbivore, and they are keystone organisms. If they are monitored more efficiently, we can identify certain changes that are happening in our ecosystem because of the many different niches that they interact with, whether it's what they eat, the temperatures they exhibit, or the animals that like to eat them, such as birds, lizards, rodents, other insects, and also the decomposers that bring the nutrients right back to the food source, which is given to the sun. The grasshoppers eat the grass that created the energy from the sun, and all of these animals eat that, and it goes right back to where it started. Unless temperatures start to increase, these grasshoppers can overpopulate the region. They run out of food too quickly, and then they move the nutrients that they take up to another region. What is a grasshopper? In short, grasshoppers are in the family Acrididae, and that's basically all grasshoppers. And you can distinguish them from their close cousins by how long their antennae are. This is very small compared to this. It's about as long as their body. Also, crickets tend to have this ovipositor, and grasshoppers do not. Grasshoppers are a good species to monitor because nearly all of their metabolic processes are monitored or can be monitored through their sensitivity to temperature. Every single life stage is sensitive and has cues to start the net and as well as their food supply. But hatching is cued by temperature, and with the irregularities in climate, they can cause misalignment with those hatchings, and they could hatch too early for their food source, or not hatch at all for a year for that matter, and then be dormant until appropriate conditions come. Also, they have increased feeding with higher temperatures. Movement can be determined by their temperature. Some grasshoppers can track their desired thermal temperature that they want to be in across long distances, and that can actually drive nutrients cycling across further distances, which will move things away from where they were initially. And also population growth, as I discussed earlier. Grasshopper variation is really interesting because there are a lot of different species, especially in the monument. There are over 17 species in the monument. Some grasshoppers are really hardy. One example is Cratopetys neglectus. It is ranged from southern Canada all the way to across the western United States, and we can find it here in the monument. Others are sensitive, and we call them specialists, and they tend to require very specific temperatures to allow them to proliferate, and that can cause landlocks, preventing their spread across long distances. One of those is a species native to Southern Oregon, and that is Chloealtis aspasma. The role of grasshoppers really lies in their biodiversity. They are excellent at finding all the different niches in the environment and using them well. And they're also very, very beautiful, as you can tell. By monitoring the grasshoppers, we can monitor the ecosystem, and that is why I set out to pilot this experiment and monitor a long-term program to where we set out temperature loggers and to understand the temperatures they're going to be exhibiting. And then hopefully, as we build data up year by year, we can exhibit some correlations between the cataloging that we will also be doing, because sometimes grasshoppers might not hatch if conditions aren't met for them to hatch. So they might skip a year and then hatch. And then so we might not have seen all the grasshoppers that could be at the sites that we monitored. And these are the sites across the monument that we monitored: Hobart Bluff Trailhead, Shale City Vista, Shale City Meadow, and way over here, Surveyor Mountain. And everything inside this green outline is the Cascade-Siskiyou National Monument. And these are a depiction of the sites. And this is me, this is Jacob Fernley, and this is Tayla Moore. And they helped me sample some of the days Tayla came out, especially in the beginning. And then the rest of the days, we found Jacob, as a young student here, and he was very helpful in helping me sample some of the grasshoppers at each of these sites. And one thing I like to point out is that all of these places have different ecological conditions. Some of them, the Shale City sites, have relatively similar altitudes, but Hobart Bluff and Surveyor Mountain are vastly different between that. Hobart Bluff being the tallest. And there's very little vegetation here. We found some in wild ginger that we think that the grasshoppers were mainly eating in that site, which I thought was pretty unique. So we went field hopping across all the different sites for 13 weeks, and I swept 200 sweeps at each site. We placed thermal data loggers in the beginning, and we collect them later on, and we're still processing that data. There was over 30 data loggers, and it's been a long year. And over 2,000 grasshoppers were collected across these 13 weeks. The most abundant grasshoppers are right here. They all happen to all be in the exact same tribe, Melanopilinae, sorry. And the one thing that is really interesting about them is that they all pretty much look alike, especially in the beginning. And before we actually knew what they were, we had to do a lot of research to distinguish them, because the primary feature because there's a lot of variation within grasshoppers, even within a species, so they can start to look like other grasshoppers. One variation is their sex organ, which is the cerci, and you can see that right here. And you can kind of see it right here. They are vastly different. This one is very large and hook-like, and this one is sort of like pointed in a way. And the other one is more bulbish, but this is a female, and we don't have very good pictures of the cerci of the males yet, unfortunately. But yeah, Melanopus ascensus was the most abundant. It represented over 50% of the grasshoppers that I collected. Odontelinotis enigma is the next, and it was significantly lower, less than 10%. And just below that was Melanopus goodeni. This is the grasshopper biodiversity varied over time. This is the number of species per 200 sweeps, and these are the weeks across time that we measured. In the beginning, this is adults. It's very hard to distinguish the nymphs, the juveniles, because there's a lot of variability, especially when they're younger, depending on food source within species as well. And so color is not very good indicator, and they're also very, very, very small. So these are the adults. There were very few adults, I think maybe just one. And then as we go into the weeks, you see more and more different species throughout, and it sort of tapers off between all the different species that we saw across the entire monument. Here is grasshopper diversity varied over space, and these are all the different sites that I measured. And this is Hobart Bluff. Shale City Meadow, Shale City Vista, and Surveyor Mountain. Notice that Hobart Bluff has the highest species richness. So that there's the most species found constantly throughout the weeks that I measured. And I propose that could potentially be part of the highways that I was talking about earlier underneath the power lines, because power lines go right through right through Hobart Bluff. And that gives access for a lot of grasshoppers and potentially many other insects and animals as well to just go right through if they like that habitat. If you notice that Shale City Meadow had the lowest amount of species, and I'll get to that later, and the others were fairly consistent and similar throughout, with different species to their site. This is grasshopper abundance varied over time. And here we have the number of individuals per 200 sweeps and the weeks across time. And these are juveniles and these are adults. As you notice, the juveniles are high in population across time, and then it tapers down to very, very, very few. We think that some grasshoppers just emerged late or something, but we did find a few till the very end. And adults, we see a number of adults, and then it inclines, and it peaks, and then it sort of plateaus. And that could be how the ecosystem is monitoring with the high abundance. Maybe birds can see a lot more of them, and then when they have low abundance, maybe that's when they're starting to breed, or it's colder, and we're just not able to catch them because they are ectotherms and they're responding to the cold. So that means they're not even jumping as much, which we did notice that because they were there sometimes, especially at the end, but they just were not jumping no matter what we did. And yeah, and this is grasshopper abundance varied over space. And this is the number of individuals per 200 sweeps. And this is Hobart Bluff, Shale City Meadow, Shale City Vista, and Surveyor Mountain. And notice that the most grasshoppers we found consistently was at Shale City Meadow. However, that had the fewest amount of species. So the species that was dominating there was Melanopus ascensus. It was dominating across all of the different sites. It was a very hardy species. And it is just so prevalent. It's really interesting because they're sort of a funky type of species, and you'll see why in a bit. So these are many of the different species here. Just want to point out all the different biodiversity. These are in one species, and see how color is it's just random. We don't know too much about what creates what creates the colors and patterns for grasshopper for every single species. It's very different across the board, and that's why we need more research. But one reason that I like to point out is that with the tribe species that were the most abundant, they were colored differently, similar to that, but they were completely different species. However, these are the same species. And right here, I'd like to point out that the most abundant species have these spurs right underneath their throats. They look like just these little bumps that are sitting there. And that's one of the diagnostics to find a subfamily that they're in. They're called Melanopolinae. So the field observations that I really liked were the egg laying of different species. They had different locations that they'd like to lay their eggs. This grasshopper is laying its eggs inside of wood, which would buffer it in a different way than, say, the crevice of a boulder. And this grasshopper is really cool. It was at Surveyor Mountain, and it was the only grasshopper in the monument that didn't have any form of wings at all. We looked underneath its little armor flap right here. This is called the pronotum. And underneath that flap, there's supposed to be wings there at least, and there was no wings, and we thought that was very interesting. And this is another grasshopper that's laying its eggs inside of dirt. And it looks like it's on a rock there, but they're just like little rocks. It wasn't a big boulder. And I thought that was pretty interesting as well. And this is a very interesting video that I made together, and I'm going to give it to you twice. See, that was a grasshopper that was mating, and they were able to jump. And this is competition between the grasshoppers, telling him to back off, raising his legs. The only grasshopper species to do this. And this is Melanopus ascensus, the most abundant grasshopper. And it looks like it's in a comatose state, but look at those legs. They were kind of hopping. And it's just sort of dangling there. Several of them I saw doing this. And again, sometimes it can be kind of fast. They're really powerful jumpers. And then it disengages when it feels safe. Right here, the legs are jumping on the male, or trying to jump. It's weird, but the moment I watched it, and the moment it detached, it went right back to normal, like nothing happened. It was very interesting mating behavior. So the conclusion and further directions is this year I cataloged 17 different grasshopper species, and more are counting. There are some species that we're still trying to find out what they are. And there's potentially more species that could hatch that didn't hatch this year, or migrate in that were further away this year. So more research is needed. Continued research is needed to understand the biodiversity and how that varies, and how we can use the grasshoppers to understand how climate change is affecting the monument. Thank you. Is there any questions?
Hi, Trevor. Great presentation. Thank you. That was really good. I'm curious about you showed a picture of the slant-faced Siskiyou grasshopper. You mentioned that it's endemic.
Yes.
I'm curious if you could speak to what would cause a species to be endemic to the monument? Is it the food source, or is it what causes grasshoppers to have such small ranges? That's a cool looking critter. Yeah.
Yeah. So it's sort of random. Evolution is not very particular with what happens, with it just sort of whatever ends up fitting, like the genes that grasshopper has that are different, it decides that this is the more optimal climate for it to live. And because of the nature that ends up being is it sort of gets stuck there through the like a geo landlock. And one of the hypotheses for many of the grasshoppers, especially the tribe of grasshoppers that are most abundant, is that during the Ice Age, there were regions called refugia, basically sanctuary for grasshoppers and other organisms, and it allowed them to differentiate. And so they would sexually differentiate, remaining very similar to what they were before, but their sex organs, the cerci that I showed you earlier, really allowed them to no longer mate, or at least as far as we know, with the other grasshoppers in the area. And so, like, if they ever met their close cousins that were separated before the Ice Age came in.
Oh, thank you.
Can you explain the term tribe species?
So the tribe is sort of like this sub family. It's not like immensely recognized as far as I know, and it's lower than the subfamily, which is Melanoplanae for spear-throated grasshoppers, which is and so the spur-throated grasshoppers would essentially, when they differentiated, a certain species of spur-throated grasshoppers were in the monument, and then at the time of when they were allowed to differentiate with the Ice Age, like whatever they differentiated to were in the same tribe, essentially, at that time period.
You mentioned that and showed in your data later in the season, you didn't see the grasshoppers jumping, so you weren't able to catch them and swiping. Do you think that you would maybe need to like change up your, or would there be potential to change up your sampling method somehow?
Yes, and that, in fact, we talked about a few ideas, maybe somehow getting them to show up either with similar sounds of stridulation, which we don't know if that would work, but also maybe like a trap where they would kind of get lured in the area. But we're still looking into understanding that. But at the same time, if we do it the same way, it tells us something about the environment. It tells us something about how much they're able to move at that time. So, and also if we depict that in our notes, it gives us some more feedback. If there's no grasshoppers there at all that we don't see in the grass, that's already dead at that point, then that's something to be said. But there were a lot of grasshoppers just not jumping.
Just last question.
Sorry if you covered this earlier. Are some of them very specific in what they eat, or are they mostly generalists?
Well, a lot of grasshoppers are generalists. They eat, but it's really hard to just say that because they differentiate so well. As far as I could tell, the biggest generalist was the most abundant one. It was at every single site, regardless of altitude, regardless of thermal extremes that they can bounce around to regulate their temperature. And so it seems like they also had different food supplies at each site. So they would just eat whatever was available to them and maybe even outcompete some of the other grasshoppers in certain sites like Shale City Meadow, where they had very few species richness, but a high abundance primarily because of that one grasshopper.
All right, folks, we're going to go on to our second speaker. Like I said, I've been working with Tayla for about 1.5 years now. Tayla isn't writing a novel, but she does bake a mean sourdough bread. Among her many talents is catching grasshoppers, baking bread, and especially finding one in particular grasshopper, Chloealtis aspasma, which she's going to be talking about today. So please join me in welcoming Tayla Moore.
Okay, so thank you, Dr. Youngblood, for that introduction. Hi, everyone. I want to thank you all for coming, both in person and through Zoom. I really appreciate it. I'm excited to discuss what I did this summer. So yeah, let's get into it. So my name is Tayla Moore. And for this summer, I wanted to uncover the natural history and geographic distributions of the Siskiyou slant-faced grasshopper, Chloealtis aspasma. So first, I wanted to talk about why grasshoppers are important. So grasshoppers are dominant herbivores in many ecosystems. Grasshoppers help maintain balance in the ecosystem by acting as an important food source for many birds, reptiles, and small mammals. Grasshoppers also provide nutrients to soil and support plant growth by nutrient cycling. And since grasshoppers respond strongly to temperature, they are good indicators for the effects of climate change. Now that I kind of discussed why grasshoppers are important, I want to discuss a genus of grasshopper, the slant-faced grasshopper. So slant-faced grasshoppers typically live in open fields or meadows at high elevations. Their dispersal ability is restricted by their lack of wings and their preference for mountainous regions at higher altitudes. And as you can see, the slant-faced grasshopper Their wing doesn't reach past the end of their abdomen, whereas the non-slant-faced grasshopper, which is this is a banded wing grasshopper called Camnula pellucida. Their wing reaches past the length of their abdomen. Some other features that differentiate slant-faced versus not slant-faced is slant-faced grasshoppers obviously have this straight edge kind of angular face, whereas the non-slant-faced have a more rounded face. And a grasshopper starts out as an egg. Once it hatches from the egg, it turns into a nymph. And following 5 instars, they turn into an adult. And adult grasshoppers lay their eggs during late summer or early fall, where the eggs can hatch the following spring. And here's a species distribution map of the genus Chloealtis grasshoppers and the 5 species. So first there's Chloealtis abdominis, this green point. You can see all these green. Chloealtis aspasma, it's this pink purplish point. Chloealtis conspersa, these blue points. Chloealtis tenae, these red points. And Chloealtis gracilis, these black points. So as you can see, Chloealtis abdominis and Chloealtis conspersa, the green and blue points, have the most widely distributed range for this genus, whereas Chloealtis aspasma, Chloealtis tenae. Oh, oh, good. Okay. Chloealtis abdominis and Chloealtis conspersa have the most widely distributed ranges, whereas Chloealtis tenae, Chloealtis aspasma, and Chloealtis gracilis are predominantly found along the west coast of North America. And highlighting Chloealtis aspasma, you'd see they've only been documented to be found in Southern Oregon. But they could potentially be in Northern California based on species distribution models, a prediction one. So yeah, only Southern Oregon so far. So we know that Chloealtis aspasma is rare and endemic. Like I said, it's only found in Southern Oregon and probably Northern California. The Bureau of Land Management and the United States Forest Service has listed this species as a species of special status. And you can see how the species can be identified by its features. So on top, the top photo is of a male, whereas the bottom photo is of a female. So you can see this species has more flat antenna compared to other species within this genus. They have a diamond-shaped nose, which you can't really see well in this photo, but they still have it. They have orange legs or an orange tibia. They have short wings that don't reach past the length of their abdomen. And most importantly, they have a slanted face. So in a 2008 survey, they found that 15 out of 44 locations that they surveyed had Chloealtis aspasma, and 5 of these sites were within the Cascade-Siskiyou National Monument. So this is a figure from the 2008 survey. The red dots are the sites where they searched, but Chloealtis aspasma was not present. Whereas the green stars are sites that they searched where Chloealtis aspasma was present. And an important part of my research was to help promote an understanding and appreciation for the Cascade-Siskiyou National Monument. So the Cascade-Siskiyou National Monument was founded to protect a wide variety of animals found within the park, including Chloealtis aspasma. Scientists know very little about the distribution and natural history of this rare and sensitive species. So to address my knowledge gap, my project will achieve the following two objectives. So my first research objective was to one, map the distribution of the Siskiyou slant-faced grasshopper, Chloealtis aspasma, and two, uncover the natural history of the Chloealtis aspasma. But to do this, we first had to find the grasshopper. So Chloealtis aspasma was first observed on August 3rd. We did this by regular weekly surveying at 4 core sites, which is Hobart Bluff Trailhead, Shield City Meadow, Shield City Vista, and Surveyor Mountain. And this is a photo of the Hobart Bluff Trailhead. Very beautiful. That was one of my favorite sites we went to. And yeah, we did regular surveying that let me determine when Chloealtis aspasma first emerged. So I could do broader surveys to locate more populations. And I could explore new regions within the monument to search for where the species may be present and potentially find new populations that haven't been documented before. So the first objective was to map the distribution of the Siskiyou slant-faced grasshopper. So my project used a preliminary species distribution model created by Dr. Youngblood to predict where the species may be found. So we visited 26 sites within the monument and searched for C. aspasma during its active site, which was in August. And this design enabled me to see if the model was accurate and to locate additional populations for this grasshopper. So here's the model. You can see this is the longitude. This is the latitude. This is Ashland. And then here is the boundaries within the Cascade-Siskiyou National Monument and the habitat suitability. So the green areas depict the most habitable areas for the species of grasshopper. And then it gradually decreases to orange to yellow to white. And then the white areas or the yellow areas are the areas where this grasshopper is least likely to be found. And as you can see, the model did a pretty good job. So out of the 26 sites, 10 of the sites is where Chloealtis aspasma was found, which is the blue circles. And 16 of the sites did not have Chloealtis aspasma, which is the red circles. And 50% of these sites, or 5 of these sites with Chloealtis aspasma, were previously undocumented. So they were new populations that haven't been discovered before, or they weren't documented before. And so my first objective was complete. I mapped the distribution of the Siskiyou slant-faced grasshopper. Now I wanted to try to uncover the natural history of the Siskiyou slant-faced grasshopper, Chloealtis aspasma. So after we located species within this genus, we used field observations to understand the feeding, thermoregulation, and reproductive patterns. But unfortunately, observing this rare and cryptic species was very hard. So I'd like you to take a look at this figure and try to find a grasshopper in there. So looking at it now, I'm pretty sure I'm the one who took this photo and I can't find it right now. So, but despite that, we were able to record the first ever video of the species stridulating. So I'd like to show you that now. Cool.
I don't have audio for some reason.
Okay.
We're
Well,
good,
yeah, it's okay. So I'll just describe it. So it's male Chloealtis aspasma stridulating in the field. Stridulating is where the grasshopper rubs its hind legs on the veins of the tegmina to create this noise. So the males make this noise to attract the females, and you can use this noise if you heard it to try to find more species. Or more, yeah, more individuals.
Were you able to mimic the call at all?
I'll take
Mimic it, it's like a clicking noise kind of, so I couldn't.
I mean, did you, call them in the field with an approximate sound?
I would speculate that you would be able to, yeah. So, I'm after distribution, but uncovering the natural history for this grasshopper is still a work in progress. So in conclusion, the species distribution model can be a very effective tool for locating additional populations of the Siskiyou slant-faced grasshopper, as well as other species. So we were able to locate 5 additional populations of grasshoppers within the monument. And so to manage these grasshopper populations, more information is needed on their natural history. So yeah, thank you for listening. I really appreciate it. And I will take any questions.
You said you're pretty sure they could be in Northern California, but they haven't been spotted in Northern California?
No, there's been no documented observations of this species in Northern California.
What would you hypothesize?
Mm,
Maybe?
well, it could just be due to people not finding them. They could still be in there, but there just hasn't been any documentation to determine that they're there.
We've been calling them short-horned
Mhm.
grasshoppers in our marketing and promotion. Is that wrong?
Well, for more accurate, under like name. I mean, it has been correct, but for a more accurate name, all grasshoppers have shorter antenna, so it's like, yeah, slant face is more accurate for, like the name. Yeah.
You used the term that they were the most, grasshoppers are the most dominant herbivores.
In most ecosystems, yeah.
So is that in the amount of food they consume or just the numbers of animals, or how is that? What are they dominant over?
Well, grasshoppers, they're, for farmers, they're pests. So because they eat a lot of grass and they kind of, like Trevor was talking about, kind of like the cattle, are kind of, they're kind of in competition with cattle because they eat so much grass. Yeah. Yeah.
How many, you might not know, this, how many individuals of like a certain species would be in like a, specific meadow that you went to, were you talking like? Hundreds?
How many different species?
How many individuals?
Oh, individuals? Oh, thousands and thousands. I mean, it depends on the location, but Hobart Bluff in the beginning they were everywhere. So I couldn't tell you how many, could be millions within a site, but yeah.
How many individuals of Chloealtis aspasma typically?
Oh, I don't know. I don't think we really counted. Well, I mean, we did, but I don't have the numbers off the top of my head. I'm sorry. Yeah.
One thing I was gonna say is, Trevor, you guys probably know this, but the Hobart area grazing was eliminated there at least 15 years ago. And on the other areas, not, so,
Mhm.
the other areas are new to the monument. So the protection has been different.
Oh, wow.
Nothing.
Yeah.
I have one more. This is kind of based off of what like Trevor was saying to me, but you, it's like that, when it's hotter, they're more active and they reproduce more quickly, or there's like a surge in their population numbers? Is that-
Yeah, so once it becomes, I'm pretty sure the number was 68 degrees Fahrenheit. If it gets hotter than that, then they begin to become more active. But if their body temperature reaches 118 degrees Fahrenheit, then they'll die. So yeah, so as long as they're in like warmer temperatures, they'll do very well. Yeah.
I was wondering if you knew when the species distribution model used, and if you could alter that species distribution model, what else would you put?
I'll go back to that. Yes, so the species distribution model, so we would add our data to kind of expand on this to make it even more accurate. Already it was fairly accurate. It did a pretty good job at determining where they could be, but adding more of the occurrence data that we collected would further make this model more accurate.
What were the most significant terms in the model? Was it elevation or a certain type of vegetation?
I think it was a combination of occurrence data as well as climate data. Yeah.
What is the interaction between grasshoppers and migrating butterflies at Hobart?
Oh, I have no idea. I don't know anything about the butterflies. Yeah.
They're probably just friends.
Yeah, they're very good friends. Yeah. Yeah.
If we wanted to learn more about grasshoppers, what would be a book you would recommend? Are there any small field guides that are kind of in the area?
There's some that are, I'm sure there's some starting to become like in development, but like I was talking about the 2008 survey done by the Bureau of Land Management. It does a really good job at discussing the species and their identifying factors, as well as where they're found. So yeah. Yeah.
You have to have them in hand to identify them easily, or is it really?
Well, it would, we could spot them in the field, but for more accuracy, I would recommend catching them with a net and then looking at them up close. But yes, and then yeah, we release them afterwards. Yeah.
My question piggybacks off Daniel's. Do you feel like difficulty in identifying, maybe just the fact that they're not used to seeing them there is why they haven't been spotted around Lake Wairega or Shasta?
Well, they are a cryptic species, so they're hard to find. Like it's hard to spot them in the field. Like the picture I showed you, like
Yeah.
it's very hard to
They
detect
don't
them.
stick out, right?
No, not at all, really.
Folks were asking about the modeling there at the end. I thought we had this slide in there. So we built the model. The 4 climate variables that were in the model were minimum temperature of coldest month, mean temperature of warmest quarter, and precipitation of driest quarter, precipitation of warmest quarter. So yeah, I'd be happy to talk more about what other things might be good to include in that. Thank you.
Just gonna introduce Hillary Rose Dawson here really quickly. Hillary is a PhD candidate in biology at the University of Oregon, and this is actually her 2nd year of receiving funding through our Friends Research Fund grant. Working with her advisor, Dr. Lucas Silva, she has been expanding on her previous research to create a preliminary list of truffle species found in the Cascade-Siskiyou National Monument. And she uses a truffle dog to find these truffles, which is just wonderful. So I will let Hillary take over from here.
Thank you very much. Hi, everyone. I'm Hillary Rose Dawson, and As Zainab said, I'm going to tell you today about the truffles of the Cascade-Siskiyou National Monument. Quickly introducing myself and my team. As she said, I'm a PhD candidate at UO. I'm also a postdoc at the Australian National University. So I'm coming to you from Australia right now. And my research focuses on plant-soil fungi interactions. This work would not be possible without my sister, Heather Dawson, who's a master's student at UO. And she's absolutely amazing with dogs and fungi and got me into this whole world of exploring underground and the interactions between the plants and the fungi. And Heather is an amazing dog trainer who's trained up Rye, a 3-year-old golden retriever, who finds all the finds that we're going to present today. What are truffles? Truffles are hypogeous or underground fruiting fungi. They're pretty much fungi that have evolved to go below ground in order to conserve moisture and be less exposed to the elements. So some of them you can see the residual classic mushroom-shaped form, like in the top 2 photos. You can see a little bit of what the stalk would have been. And then some of them at this point have lost that residual form, and they look purely like nothing else you've seen. So masses of spores like the Elaphomyces on the lower left, or just the intricate folds of a tuber on the lower right. And they have evolved to be found by animals that eat them and spread their spores this way. So we're going to think a lot about the odor of these truffles as a key characteristic. You can find truffles by going out there without a dog, but it's really difficult, especially for those of us who've experienced truffling with dogs. So when we go out to find truffles, we've got Rye with us, and we come to a new site, we tell him truffle, we prime him with showing him the ball that he really wants, and we just tell him, go find a truffle. And he takes off through the forest or the open grasses, wherever we are, looking for that odor with his amazing nose. He's constantly searching. We're just following along until we see him dig, and then we stop and wait for him to get deep enough that we can see the signs that he's found us a truffle, and we excavate it before he picks it up. And well, he's not trying to eat it, but he's trying to show us where it is, which can be a little bit damaging for the truffle. And then we give him his reward, which for Rye, he wants more than anything us to throw his ball. So that's his reward. As I mentioned, a key to the truffle world is odors. So if you've heard of culinary truffles, like you might put on a very fancy meal, those have a very distinctive truffle aroma. What we're looking for include all sorts of different aromas, and Rye can generalize across all of these, which is, we're still not entirely sure the biology of that and how he knows how all these fungi that are not necessarily related in a phylogenetic sense are what we're looking for. So some of them smell like artificial banana or like burnt brakes, shoe store, carpet glue, fennel. It's a really interesting process trying to figure out how to describe the aromas that these truffle finds present. And here's a short video of what it's like to be out truffling with Rye. This is for the culinary Oregon Blacks. So this is a commercial forest, but it's the same process when we're doing it for the diversity of truffles as well. So he's located a general spot, he's digging, Heather's getting closer, and she's like, yep, there's the black truffle. Yep, right, there's your black truffle. She's going to admire the truffle, but Rye doesn't really care about it at this point. He's just waiting for his ball. It's like, yeah, okay, truffles, done with that. Ball? There we go. Now we get to have a ball. And that's what makes him really happy. So for our surveys, we go out and we do a lot of different forays in Monument. We're trying to hit different times of year, different locations. For each one, as I described, we're going out with Rye, searching the area. Then we mark each truffle find with a GPS location, document which truffle it was, and if it's a species that we haven't analyzed before, we collect the truffle itself, dry it, and collect some tissue off of it for DNA analysis. We're running a packed bio-DNA analysis, which is more regions than would ordinarily be done using historical methods. So sometimes this means that we can also pick up secondary fungi within the truffle, and we've got a higher chance of getting an accurate match to other truffles in the databases. As mentioned, we had a grant from the Friends last year, and we went to 10 different sites, 3 different times of the year, and we found 27 species, many of which were not described. So we were super excited. This is high diversity, and we really wanted to go back. So I reapplied for another year of funding, and we've now managed to visit almost 30 sites. We made twice as many visits in the past year, and we've just tried to go all different places in the monument, particularly hitting unusual habitats and places that we hadn't been before. We've been focusing on the conifer forests of the higher elevations, and we've also been looking at the oak savannas in the lower elevations. We chose these habitats because these are the trees that are most likely to associate with truffles, and we're looking for just getting as many species as possible while we have the chance to be down in the monument. And the truffles have high host specificity. When we're in the conifers, we found 35 species that we did not find in the oaks. And again, when we're in the oaks, we found 15 species that we did not find in the conifers. Sometimes we do go to areas that are mixed between conifer and oaks, but then we can't say whether a species was associated with a conifer or an oak, so those haven't been counted here. From our DNA sequences so far, we've got more DNA to analyze from our most recent surveys. We've found 57 unique species. Almost half of those species do not have a match in a major international genetic database called GenBank. And there's a very high diversity of truffles. This is 26 different genera, which is just incredible. Over his lifetime, Rai has found us about 45 different genera. So that gives you a scope of how many truffle genera we've seen. You can see here that we've got all the different, we've given the different genera, the different species that we found so far this year, colored in different colors, so that you get a sense of the distribution of all these different genera. There's a lot of hysterangium, but you can see that they have they don't actually say which species they are precisely. They just say CS134 or CS63. And that means that we don't have a genetic match in GenBank for these DNA results. So possibly we have an undescribed species here, or maybe we have a species that's already documented in an herbarium, but no one has done the DNA analysis yet. So we can't match those two things up without a lot of detective work. So this is a lot of truffle diversity, and I want to show you some pretty pictures. This is Lepiotic verdiglyba. It's really that blue, genuinely a blue truffle, which is just so odd. And it has a strong aroma of bubblegum and almonds. And this one was such a cool find because it's only known from 2 other sites in California. It's a super rare truffle. However, where we found it in CSNM, it was pretty abundant. So is this an example of something that's not documented because it's hard to find fungi that are underground? Or is this something that is really specific to a particular host or a certain habitat? Then there's Hydnotriopsis sechelii, which we found in oak habitat. It's from the cup fungus family, Pizzaceae, and Pizzaceae has a fair number of truffles, but most of them grow in the desert, not where you expect up here in Oregon in the oaks. This one was also an example of how truffles can really trick a researcher, even when we think we know what we're doing. There's 2 major different ways of looking at fungi. There's So most fungi divide out into two different major groups, the basidiomycetes and the ascomycetes. And with Hydnotriopsis, it felt like a basidiomycete truffle. It wasn't firm, it was a little bit squishy to the touch. The structure, everything sort of indicated a basidio. But when we put it under a microscope, which is how you tell the two apart, we saw that it had asci, or the little sacs around its spores. And so it's an ascomycete, even though on the first go, it looked like a basidiomycete. And I really noticed that about studying truffles is they keep us guessing. There's so many unknowns out there. Another cool find was Sedecula pulvinata. This one looks a lot like a puffball, but we found it underground and it's not even closely related to a puffball. It's actually most closely related to that thin crust of fungi you might see if you overturn a log and look on the underside. So the inset photo here, that's a crust fungus that's in the same family as, or likely in the same family as the Sedecula pulvinata. This was thought to be the only crust fungus family truffle until we uncovered this one, which DNA analysis tells us is in the same family and is in fact in the genus that the family name is called after, Coneoptera. It didn't have a clear form. It was just sort of a weird mass underground, but it's definitely a truffle with a clear truffle aroma that rye scented, which tells us that this thing has evolved to be eaten by animals. So we now have two truffles in this family, and as far as we know, this is the first time this species has been found. Then there's the true truffles. This is the genus tuber, and this is the one that's so well known from the culinary world. But what people often don't know is tubers go so far beyond just the culinary truffles. These are 6 species that we found in CSNM. We found this genus is across CSNM pretty broadly. It's fairly abundant, but there's a lot of different species. And even though they're related to the culinary tuber, you probably don't want to eat most of them. These are the ones that can smell like carpet glue and shoe stores and just that really acrid aroma. We found them with both conifers and oaks. The name tuber quercicola there on the upper right, that's saying that it is with quercus or oaks. And the one in the center we thought was tuber quercicola, but again, DNA surprised us. It's not. It's a whole different tuber species that so far we only have documented in CSNM. And finally, there's truffles with fungal parasites. This is fairly unusual. We've been truffling for a few years now and only found a handful of parasitized truffles. So it's been surprising to find 2 different examples of this in CSNM. On the left side, there's a Quaramyces truffle that's been parasitized by what's likely Melanospora. It didn't come out in the DNA, so we are not certain, but the spores match Melanospora. And Melanospora is known from tuber parasitism, and Quaramyces is related to tubers. The Quaramyces spores looked perfectly healthy though, so likely this parasite was not negatively impacting the Quaramyces ability to reproduce. Then there was also a parasitized Balsamia, and that was parasitized by a Microascus, which is a filamentous fungus that has a broad range of forms, including you might find it if you just sample your soil, and it's also an animal disease. And here it's parasitizing Balsamia. So lots of different ways to be this one type of fungus. What are we going to do now? We're going to finish sequencing and finalizing a list of truffles for CSNM. We're going to be responsible with our data and deposit the sequences in GenBank so that other researchers can use these data as well. And then we're going to also put the specimens in the SOU herbarium for future discoveries. Finally, we hope to publish a paper describing the truffles of CSNM, which is also going to be a paper documenting as a case study how truffle dogs can be used to increase our knowledge of underground fungal diversity. I'd like to acknowledge everyone who's been a big part of this. First, the Friends of the Cascade-Siskiyou National Monument, thank you so much for 2 years of funding. This has been so much fun. We've found so many cool things and we're super excited to share them with the world. Lucas Silva and Biddy Roy are our mentors at the University of Oregon, and they've really supported and encouraged our research along the way. Scott Loring has helped us find the best truffle spots at CSNM and helped us get access to those. And Jonathan Frank has come out with us in all sorts of weather to go find truffles and share in that excitement and share his knowledge. Carolyn Delovich at the University of Oregon is our bioinformatics wizard. Without her, we would have a lot of data that we could not understand. And then finally, the photographs of truffles are all by my sister, Heather Dawson, who has trained Rye and also made this possible. Thank you so much, and I'll take any questions.
Hillary, can you hear us?
I can just barely hear you. If you speak loudly.
Okay.
Where's the microphone?
It's over here.
I think you got people. That's gonna let them
Here, I can
Do
try.
you have a theory on the various smells? Are they simply waste products, or are they metabolites made at some expense for defense?
I don't know what the origin of the odors is, but they serve a clear evolutionary purpose. Without those odors, animals wouldn't be able to find these fungi underground. So they're sort of a key to get mammals to dig, beetles to burrow, anything to come eat this ripe truffle so that the spores are ingested. And then the animal walks away and poops out those spores, helping the truffle spread its range.
You find a very different kinds of truffle in one location?
Yes, we found, oh goodness, 10 or 12 species, I think, in our most diverse site. Sometimes we'll find only 1 or 2, but there's a lovely fir site up in the monument that has held the highest truffle diversity of anywhere we've been.
How long did it take to train Rye?
Rye's a special case. Heather took 2 years training her first dog on culinary truffles, and then she got Rye. So Heather's first dog, Cricket, was sort of able to help train Rye because she would find a culinary truffle, and then we'd pull her off and show Rye, okay, dig here. You're going to get a reward when you show us where that truffle is. Then it was sort of his own idea to start moving away from the culinary truffles and into all the other truffles that were out there. At the time, we didn't really know how many truffles existed in the Pacific Northwest until he just kept finding more and more of them. And we have to go out and go, okay, what have we just found? Who can we find that can identify this? I think he found his first truffle around 6 months, but he started diversifying at about a year.
Do you have a hypothesis of why there are so many unknown to science or potentially unknown to science truffles in the monument?
I think the high diversity in the monument is because it has the flora of California and the flora of Oregon overlapping. So you get some really interesting plant interactions there, which if we assume the above ground diversity is interacting with or driving the below ground diversity, probably means that we have a really high diversity of the 2 fungal floras coming together as well. And then they're undescribed because no one's looking for them, no one's documenting them. Some of them, it's really, really hard to find them without a dog. Like one of Rye's easiest to find genera is Genia, which is maybe the size of a pencil eraser and the exact same color and shape as a lump of dirt. So if you don't have Rye picking it up and saying, that's a truffle, good luck finding it. So I think it's a combination of just high biodiversity in the monument overall, combined with the difficulty of finding truffles and documenting them, and getting the funding to look at the DNA analysis to go into, okay, what is a species and how can we differentiate these?
What wild animals eat truffles?
One of the most known ones is our flying squirrels and the redback voles. You can also find a fair number of beetles in millipedes. There's a whole section of millipedes that are focused on eating fungi, and we find a bunch of those in the truffles. Pretty much anything that can get their hands on a truffle will eat a truffle, including humans. Although most of the ones that we find in CSNM you don't want to eat because, my goodness, rotting onions are a terrible odor.
So with the diversity in the smells, how do you think the dog picks up on all the different diversity that they're if he's using smell to find these, is there potentially a similar chemical in all of them that is just overwhelmed by the other chemicals that it's releasing?
It implies that there's a common odor in between all of them because rye doesn't scent for non-truffle fungi. So he's got something going on other than just, this is a fungus. Sometimes he will scent for mycelium mats that are just sort of loose fungal material growing underground, but they always have a strong odor to us as well as to him. Whereas some of the truffles that he finds have no odor to us, but a clear odor to him. And it's a bit of a mystery. And if I knew how to do work on odors, I think that that would be a fascinating project of just analyzing all these different aromas, looking at their biochemistry and seeing what the commonalities are.
Any other questions? No? Well, thank you so much.
Thank you, Hillary.
It was so interesting to see how her research has progressed after a second year of funding.
She wanted to start here.
Right?
How many?
She can fly over here from Australia and visit the monument. Well, thank you so much to all of our wonderful presenters and all of you for being here and for your great questions. There's so many great questions. Thank you for that. I would like to remind everyone that the Friends is funded off of donations and grants. So if you would like, you can donate via cash, check, or charge. We have envelopes up at our merch table, or we also have somewhere on our website that you can donate as well if you are inclined. And we also have some merch for sale up there, some really cool t-shirts, hats, little bumblebee guide. Go out there and try to identify bumblebees. But yes, again, thank you all for being here. And feel free to take some more food when you leave. And this presentation, as you all heard, was recorded. So we'll have this back up on our website. So if you want to watch it again, or if you want to share it with someone who wasn't here, that will be available. And I'll email it to everyone who registered on Eventbrite, but that will also be on our YouTube and on our website. So yes, thank you.